Hello everyone, this is Daniel Blackburn. This is our third online lecture after the suspension of classes due to the COVID. Today's lecture is going to be about soil archaea, phages, and soil microbial interaction. This is part of the course SWAE3411 Environmental Soil Microbiology. So, talking about Archaea, we owe the classification of Archaea as a third branch of the tree of life to Carl Woese in the 1970s. So, Carl Woese was mostly comparing the genes that codes for the ribosomal RNA, the 16S rRNA genes, and he found out that the archaeobacteria and bacteria, they were fairly different from each other. And due to that dissimilarities, it was obvious that these two prokaryotes should not be classified together in the same group. So it was proposed then that we should separate archaea and bacteria in two different completely branches of the tree of life. So, such as bacteria, archaea are very, very primitive organisms that date back to 3.5 billion years ago that they are appearing on Earth. But archaea has many similarities with bacteria. For example, they have circular DNA in the cytoplasm. They don't have nuclei or nucleus. Also, archaea it possesses cell wall material, but the cell wall material it's very different from bacteria, and it's more similar to what we found in eukaryotes. Bacteria, for example, will have 1 point of replication on the circular genome, whilst archaea could have more than 1 point of replication on the circular genome. And the machinery, the molecular machinery from archaea that replicates the DNA, it's more similar to eukaryotes than that is for bacteria. So due to all of this, archaea is fairly well classified as a completely different branch of the tree of life. But this is a little bit recent, and it's not well understood fully all the roles of different archaea in the environment, because archaea are hard to culture and they are usually extremophiles. So this is showing the type of environments that we can isolate archaea, and archaea becomes more abundant. For example, thermal hot springs, volcanic lakes, very acidic conditions, or very extreme conditions with high salinity, very hot, or highly acidic. That's the places where we find the highest abundance of archaea. In soil, archaea will be about 1% to 5% mostly, depending on how anaerobic is the system. It's not highly abundant in soil, but they play a very important role on these systems, especially on the they are methanogens. Yeah? So archaea plays a really important role on the carbon cycle. So archaea are characterized for having this cell wall material, which is called the envelope. Yeah, this envelope it can form these weird shapes, flattened shapes, and the reason for that is what is the presence of what we call the S layer. The S layer is one of the main characteristics of the separation of archaea morphologically. Yeah. You can only observe this type of S layer when you have electron microscopy of these cells, not when you do normal light microscopy, because these cells are very small. They are similar to size to what a bacteria is, about 2 microns to 5 microns, sometimes below 1 micron also. Yeah. So what is the S layer? The S layer is either a form of protein or glycoprotein. It's about 20 to 40 nanometers thick, and it's usually placed outside the cell membrane. So this helps these archaea to resist the harsh conditions of the environment. One other thing that can be said about the archaea is that sometimes the lipid bilayer is not a lipid bilayer at all. It can be a lipid monolayer. So this is also very characteristic, and these peculiarities help these organisms to act like extremophiles and being able to survive in extreme environments like very hot conditions over 100 degrees Celsius, or very saline conditions, or very acidic conditions, for example. So here are some of the configurations that you can find of the cell wall and the cell membranes of archaea, and you will have that this is very variable, yeah, very variable. So you can find all these type of configurations with the S layer completely exposed, or protected by a protein sheet, or by a layer of metanochondrotin. Um, and you can have a polysaccharide layer, or you have, like in gram-negative bacteria, you will have also the possibility of having a double bilayer on the cells in a periplasmatic content. So in theory, you can also separate archaea as you do for bacteria in gram-positive and gram-negative. They can also respond to the gram staining in the same way, but the archaea is a lot more diverse when it comes to the configurations of the cell wall material in comparison to bacteria. So this is showing that the phospholipids from archaea are very also different from what is the phospholipids from bacteria. So you have this presence of phytanyl side chains on the lipid part of the phospholipid. And while in archaea you have ether linkage with on the head of the phospholipid, in the bacteria you will have an ester linkage. So this is a very significant difference between these two groups of prokaryotes. Regarding the genome of archaea, the genome of archaea is significantly smaller than bacteria, which leads to speculation that these are more primitive microbes in comparison to bacteria. And for example, in E. coli, you have 2.5 times 10 to the 9th Daltons, and in T. acidophilum, you have 0.8 times 10 to the 9th Daltons. Archaea also, similar to bacteria, has a circular chromosome, but it's very different, as I spoke before, that you may have more than one site of in which the replication can start. In the transcription and translation processes, they're very similar to what is in eukaryotes, and more similar to eukaryotes than it is with bacteria. So that is also some of the main differences between them. So in metabolism, we can say that as well as with bacteria, they can also be chemolithotrophs. Yeah? The chemolithotrophy is not exclusive for bacteria. It can be bacteria or archaea. Also, you will find that some of archaea can be autotrophs. So all the variety of biochemical processes and the energy and carbon metabolisms that you can find in bacteria, you can also find in archaea. Nonetheless, you will find that Archaea plays very important and exclusive roles when it comes to the biochemistry of anaerobic conditions. Mostly when you have highly anoxic conditions, you will have bacteria playing a very specific role in some of the nutrient cycles, especially when it comes to carbon and sulfur. Also, denitrification can be influenced by Archaea. Um, also in terms of metabolism, the glycolysis from Archaea is a bit different from bacteria. It's called the Entner-Doudoroff pathway. Um, uh, the methanogenesis is exclusive to bacteria. We will speak about that when we talk about the carbon cycle. Um, there's a specific phylum of Archaea, the Archaeota, which is responsible for methanogenesis. So the emission of methane from anaerobic conditions is something that is exclusive for Archaea. Um, so yeah, Archaea as well as bacteria also can fix atmospheric nitrogen. Okay, so this is not exclusive from bacteria. So when we talk about the mechanism, the metabolism from Archaea and bacteria, the whole variety that you can find in bacteria, you can find in Archaea. And I think the main exclusive thing that you will find in Archaea that you do not find in bacteria are the methanogens. Okay, so now we're going to talk about phages. Phages are what we call the viruses of bacteria, of prokaryotes, or archaea also. So you have viruses that can attack every single type of cell, uh, and these viruses, some of them are specialized in prokaryotic cells. These are the electron microscopy of these cells. They look like a little bit like a sort of like a spider-like. They have this tail fiber that can bind to the prokaryotic cells and then use that to inject the RNA inside the cells. So phages or bacteriophages are they are the viruses that use prokaryotes as a host. You know, uses prokaryote as a host. Viruses, in general, they are not considered living beings. They are not alive. You know, they cannot live by themselves. They cannot replicate. They need another living cell to replicate. But nevertheless, these are very important in the ecological relationships of soil microbes. And if not for these viruses, some of the populations could go out of control, and there would not be a good regulation of the population explosions of some microbes in the soil. So viruses are very important on this succession of different populations in different environments. Before we had antibiotics, the main line of research of combating specific infections, bacterial infections, was to treat it with specific bacteriophages for these pathogens. The treatments with phages, they are more specific than they are the antibiotics. Antibiotics can attack a wide range of prokaryotes, while the phages would be more specific for a single pathogen. So this is also good because when you take antibiotics, what you're doing is you're killing also your flora, yeah, your flora and other bacteria, good bacteria that is in your body. And if you if you were able to treat some infection with phages, it will mean that you will not be harming the 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 good microbes on your body. So this is a line of research that's slowly returning, and the use of phages to treat diseases. But for us, what we're most concerned about phages is that their role in the ecology of the soil, because they're regulating the population of prokaryotes. So when we talk about phages, we don't talk about a colony of phages. We talk about a plague. So this is related to how we can grow these phages. Yeah, the phages are grown over a lawn of cells. First, you can grow a cells. Let's say a phage that attacks E. coli. You have to overgrow a plate with E. coli, and then you apply this solution containing phages, and the clear zones that form on the plate because of the death of the E. coli cells, these clear zones is what we call the plaques. Yeah? And this is like a negative from what a colony would be. It's like clear zones on an overgrown plate. And you can, if you have overgrown plates from soil cultures, you sometimes find that these clear zones that could be either the presence of phages or could be also the presence of antimicrobial activity from different bacteria on those plates. Yeah. All right. So because these phages, they are infecting cells, they have a latent period where they are colonizing the cells and replicating. Then all the cells will break, and there's going to be a release period where these viruses are going to infect other cells. So every time you have this happening, it occurs in pulses. So you have the time where the virus is just growing inside the cells, and when the cells break, then you see that increase in the size of the plaque. And then because of this rhythm that is infecting, uh, producing more viruses, and then being released to the environment, infecting more cells, incubating and releasing, and these bursts of growth makes that the growth curve of uh, uh, phages, they are like a stair-like growth curve. They're increasing in pulses, like here in the figure that you see. All right, so that was all that we have for phages. So let's talk about microbe interactions and plant microbe interactions. So this is about ecological relationships that we have in soils. So the main role of phages in soil for our understanding is that they will be regulating some of the microbe interactions. Some populations that are overgrown, they can be down-regulated by uh, phages in these environments. They play a very critical role in keeping the diversity on these environments. Also, they keep a important role in the genetic diversity of these systems. Okay? But let's talk about what are the different types of microbe interaction, and how can we classify them and understand them better. So first of all, one thing it's important to mention in the soils, it's these interactions. They don't have to be constant. Yeah, you can have that some in some conditions. Some microbes could have a neutral relationship, like a neutralism, and then the conditions are so that they start competing with each other. When you have the depletion of some nutrient, for example, and you can have this competition happening. Or it could be that some population could be in a protocooperation with another organism, but might as well be just in some other relationship with other microbes. So the type of the ecological relationship doesn't have to be constant in time. It can change for the same group of microbes. But as we classify it, we have to be very clear of what we mean about that. So first of all, neutralism is when the population of one microbe is not affected and the population of second microbe is also not affected. Commensalism is when one population is not affected, but yes, the other population is benefiting. from the appearance of the other microbe. Um, Synergism is when both are benefiting. Mutualism also. So synergism is what I was calling the protocooperation before, and mutualism here is more referring to symbiosis. Competition is when both populations are negatively affected by one another. Amensalism is when one is not affected and the other one is negatively affected. Parasitism is one is beneficially benefiting from the other one. Also predation in that way. We're going to make the difference between what is parasitism and predation in a second. Um, so another way of redistributing these classifications is just putting if the relationship is negative, neutral, or positive in one side, or if it of species one and species two, and then you can distribute these relationships in a table in this way. Yeah? This is classical because this is also, as I told, this is a little bit more fluid and can be varying in time. All right. So first of all, neutralism is something that is not very common. Almost always, different populations of microbes will be interfering with the behavior of other populations. Almost never you will have that this relationship is completely neutral and one species is not affected by another species. Uh, uh, Examples of how this could happen if they are spatially separated. One is occupying the micropore, one is occupying the macropore, one is in one layer, the other one is in another layer. That could be one situation where these microbes are not affecting one another, but this is usually very rare. Commensalism is when one species benefit from another one. For example, if you condition, make a habitat more favorable for the other one. For example, you have an autotroph that is producing oxic conditions for another microbe. The autotroph is not making that on purpose to condition the ecosystem for the next species, but the next species, yes, will be benefiting from that. Other example would be, for example, that some free living nitrogen fixers could be releasing more nutrient on the environment and helping in that way other microbes with nutrients. So amensalism is one microbe is not benefited, but yes, the other microbe is negatively affected. That could be in the same way. If you imagine that some autotroph is creating oxic conditions on the soil, the microbe that is an anaerobic microbe will be negatively affected by that organism. It doesn't mean that this the first microbe that is doing that on purpose to negatively affect the other one, um, but it could be something that is a consequence of, yeah. But it could be on purpose also. For example, when a microbe is producing antibiotic in soils. So competition is the classical, the more common type of relationship that we have in soil is when, for example, you have a limited resource or nutrient source in the environment, and both populations of different species are competing for that natural resource, for that nutrient, and then of course that is depleting the amount of nutrient and the other species can use on that habitat. So because they are using the same resources, then they are competing with each other and then causing the other one to have a lower population than they would have in the case that these species were not present on the environment. So competition for nutrients is the main way that you can find this in soils, but there are also other things that could be happening. For example, conditions, competition for um, oxygen, for example. Um, so you have antagonism in soils, also competition. It's normally we talk about that for nutrients. The faster microbe, the faster growing one will outcompete the slower growing one. But antagonism is when we talk about that is purposely producing antimicrobial agents to decrease the population of the other one. Mutualism, we're going to separate mutualism in two types of mutualism. One is a synergism or proto-cooperation, when two species can benefit from each other, but they are not necessarily working together. Species A can live well by itself, species B could live well by itself, but together they benefit from each other. Um, for example, you have rhizosphere bacteria that are solubilized in phosphorus. Uh, the plant benefits from the phosphorus solubilization. The bacteria benefits from the root exudates. They are both benefiting from each other, but the plant can live without the bacteria, and the bacteria can live without the plant in completely separate conditions. But in the case they are together, they are both benefiting from each other. The other type of mutualism is symbiosis, and is usually taken as when these populations have to live together. They depend on each other for survival. For example, in lichens, when you have fungi and cyanobacteria living together, and they need each other to live. For example, some of the mycorrhiza in soil, they need the root. They cannot survive without the plant. Or some rhizobium species, they need to have the legume to be able to make their to grow and have their colonies. Otherwise, they will not live have free living on soils. Um, the next ecological relationship is parasitism, and we want to make a difference between parasitism and predation. Yeah, parasitism and predation, they are both benefiting from another population, and the other population is being negatively affected by this relationship. But the difference is a parasite. The difference is the parasite will not kill the host necessarily or immediately. It will use the host as long as it can and then move on to another host. It will extract nutrients and keep the host alive as much as possible as a source of that nutrient that it needs. Whilst when you have predation, the predator usually results in the death of the prey. And it's not a function of the size of the microbe. It doesn't mean that the smaller microbe will be the prey and the bigger one will be the predator. It could be the other way around also. Okay, but the relationship is characterized by that when this predation happens, it usually results on the death of the prey. And last but not least, we have to think about these ecological relationships. They are happening. It's not a constant thing. Yeah, it's a time-dependent thing. And every time you change the populations, you change the biochemistry, you change the dynamics on the soil, and there is a succession happening. Every relationship changes the environment, which creates the condition for the next population, which creates the condition for the next population. And then there is a succession. The most classical example of this succession is, for example, when you have the decomposition of organic matter during composting. We will have a mesophilic phase, then you have a thermophilic phase, and then you have different temperatures conditioning different types of microbes. You have pH changing on this medium, which will be conditioning different types of microbes. You have nutrient availability, carbon to nitrogen ratio starting to decrease in time in this material, and that will allow for different groups of microbes to colonize at different stages. So this succession is very well illustrated in the composting, but it's also happening in soil, and it could be happening, for example, in any sort of event, for example, after a rainfall, or when the death of some animal in the soil, or decomposition of the litter on the surface of the soil. There is a natural succession that is happening in different environments. And it's a good way of thinking about soil microbes as a dynamic succession of continuously ranging a variety of ecological relationships. So this is a very complex system, and understanding this system is quite crucial for understanding plant behavior, for example, and the distribution, the taxonomic distribution of these microbes. So the other thing that when we talk about the successions, usually we are talking about there's a high tendency of explaining that with the as a function of the stoichiometry of the system, which means that if you have different nutrients, and these nutrients are more available or less available, or they have a distant distribution in time, that will be directly conditioning what type of microbes are appearing, what type of populations are appearing. But that is not always the case. There are other relationships which are non-stoichiometric, and many of these relationships are about the bacteria communicating or microbes communicating with each other through chemicals in the soil. And there are some weird behaviors which are hard to place uh, in the classical distribution of ecological relationships. And we already talked about this cordon sensing, when the microbes sense the population of their own taxa, or they sense the population of other microbes um, in the environment. So just to mention that there are some more complex relationships and different relationships on the environment that are not directly a result of this classical distribution of the ecological relationships. Thank you very much.